Good morning, everybody. Thank you to give me the opportunity to present our platform process for AAV manufacturing that we have implemented at Viralgen. So this is the disclaimer. Viralgen is a CDMO that is located in the north of Spain in a city called San Sebastian, very close to France. We have right now two different facilities very close to each other, less than 1 kilometer. In fact, we are rotating people and sample all over the both facilities. Because it's located in Europe, we need to have the GMP accreditation, EMA GMP accreditation for any kind of manufacturing. So it's something that we have in 2019 for clinical phases, and more recently in 2022 for commercial phases. In total, we have around 300,000 square meters, and around 35,000 of these are GMP facility. It's distributed in 7 independent GMP facilities with 250, 500, and 2,000 liter bioreactor, and between both, we are sharing the QC and fill and finish facility. It's important to say that we are planning to have a facility also in the US in 2026. So we have to define Viralgen in 4 ideas. So we are a GMP compliant facility that we are manufacturing AAV only. We are not producing any other kind of virus. We have our own proprietary mammalian cell, which is called Protein cell line, and I'm going to get into that in 1 minute. We have a large-scale GMP facility from 50 to 2,000 liters, and we have a massive developmental expertise, not only in process, but also in analytical methods. So like I said, we have our own cell line, proprietary cell line, that it was derived by Youth Samulski in North Carolina. It's called the Protein cell line. This cell line is derived from the HET 293 cell line. It's a cell suspension cell line. We are not using any animal-derived components. In terms of safety, it's quite important. We're not having SB40 sequence because it was derived from the original 293 cells. And of course, it has a high transfection and production capacity. We're using a triple transfection system that we have the helper plasmid, the repcap plasmid, and then the transgene plasmid. We usually have off the cell the helper and the repcap, and then the client has to can only the transgene flanked by the ITR. With the 3 plasmid together, we put it inside of the cell and we produce the viruses. So with this system, we have been able to implement a manufacturing platform that has different steps. The first is the upstream process that is all the way to cell expansion and transfection that we have from 50 to 2,000 liter single-use bioreactor. Then we have a downstream process that is for purification, concentration, and buffer formulation that we have 5 steps, clarification, affinity chromatography, density gradient, anion exchange chromatography, and TFF. We have our own fill and finish capabilities. Until now, we have been working with semi-automatic system, but hopefully at the beginning of next year, we're going to have a completely automatic system inside of the isolator. And around 80% of the quality control that we are using for release the product has been developed and validated inside of Viralgen. No? I'm not going to explain here what is the roadmap for the commercialization, right? But from the manufacturing point of view, it's important to say that until now, most of the CDMOs has been involved at the really very beginning. So it means that we have been only scaling up all the production. But now we have several clients in Viralgen that they want to move to the next step, which is the phase 3 and commercial phases. This is extremely challenging from the manufacturing point of view, and I'm going to give you a couple of examples of the client that we are working with. So we have some clients that they are working in rare diseases, which means that they are going to have very few patients. Of course, gene therapy, as you know, is a single dosage treatment. So if we put everything together, it means that with 1 or 2 or 3 batches, it's going to be more than enough to treat the whole population, no? In the world. So it means that it's going to be extremely challenging for the manufacturing. In the other extreme of the coin, we have some clients that they are working in high prevalent diseases even though it's a single dose, they are using a high concentration for the dose. It means that we are going to have to produce many batches, no? And if we put everything together, the cost of the material that we are using and the yields that the whole field is now offering, if we put both of them together, most of these programs are going to be at risk because it's going to be very difficult to supply all these viruses. So what Viralgen has been done so far to solve all these problems? So for the first part, like I said, for those clients that they need to produce very few number of batches, it's going to be very difficult to set any kind of a specification. No? So for this kind of thing, we think that the platform approach is the right way to go. No? If we're producing different viruses, but always in the same way, we think that we can use some of this data from one product to another product, because otherwise it's going to be impossible to set this specification. Of course, for this platform approach, we're gonna have to support with a massive data set, no? So in this sense, we have been produced in the last 5 years more than 900 batches, always with the same cell, always with the same process. And we have been all the way up from 2 liters to 50 liters to 250 and 500 liters to 2,000 liters, from the reserve batches to all the GMP batches. More important, not only that we have all these batches, but we have also worked with many serotypes. All this serotype has been exposed to this platform, and now we have a clear idea how every single serotype behaves in our platform. No? As you can imagine, the amount of data that we are generating right now with all this number of batches is absolutely insane. So that is the reason that around 3 years ago, we decided to start a massive digitalization of the whole company. No? So we are really proud to say that most of the equipment, most of the process that we have in Viralgen, now we are getting data almost at real time. This allows us to compare batch to batch, even during the production, the whole production. So if we are facing any problem, we usually we are able to solve quite fast. But not only that, but also we can compare all the production that we are doing from batch to batch, from serotype to serotype as well. No? It's important to say that we have a really important IT team that they are helping us with mathematical model, that they are helping us to do all these comparisons. So we are using quite a lot to answer questions from our client or even from the regulator. So applying mathematical model is easier to answer most of the questions that our client has. No? This is when we put all this data together and we analyze everything, it is clear that this platform strategy make a lot of sense. No? So for example, just before the transfection, if we are working always with the same line, we are always working with the same media, with the same parameter of expansion, it is clear that we have almost 900 batches that we can put together and we can share with all our clients to support the expansion system. Of course, in DSP is completely different because now we have different serotypes and different GOI. But when you put everything together, you can see that clearly that the different serotypes behave different. AB9 is different than AB7 and different than AB8. But when you see AB9, you can, for example, see that all the batches that we are producing with this serotype, mostly independent of the GOI, behave exactly the same in terms of impurities that we are getting, in terms of hostile DNA, and so on and so forth. No? The same for the fill and finish. And of course, we think that, for example, validation of analytical method is time-consuming and it's quite expensive. So we really believe that if we we validate one method, we can use from one serotype to another serotype if we have important comparability studies. No? So now that was the theory. We have to demonstrate that this platform somehow it could be helpful for commercialization. That is the reason that we have recently set up our MSAT lab. The idea is that we have been able to scale all the way up to 2,000 liters. We set all the parameters in terms of transfection, mixing time, etc. So now we have to develop a scaled-down model again to be able to characterize the whole process. So we really believe that with this characterization, but with all the data that we are generating, that we have been generating in the last few years, we think that we can have a very strong package. No? But this is just the future. What about the present? So we have exposed this idea of the platform to different regulators all over the world, and with our client, we have been able to participate. We are participating right now in 21 clinical trials. 20 out of these 21 has been already authorized, and 10 out of these 20 they have been already injected in human. We have been exposed to not only to FDA but also to many countries in the UA, in Europe, and also to UK and Canada. We have a very strong regulatory team right now that set up a few years ago, set up a CMC package. That this CMC package has been exposed to the different regulatory agencies. Now, with all these questions that has been answered, now we are feeding back this CMC package. So we really believe now that we have a very strong CMC package that we can help our client to go through all the submission. So what happened in the other side of the coin, that we're going to need many batches to be produced. So the only way that we think that this problem can be solved is just to yield improvement. This is something that has been done in the antibody world many years ago, and this is something that the gene therapy field needs to face really fast. No? Right now, we are not able to produce enough virus for those programs that we have a high prevalence diseases. No? In this sense, together with ASBio, we have been developed what we call the next generation process. So we have been moving out from the typical fed batch expansion system to a new perfusion system. So the idea is quite simple. If we can increase the number of cells because of this perfusion system in the same site and in the same volume, we will be able to increase the yields of the virus that we are producing. And even that the density gradient at the DSP is working really well for full empty separation, we do understand that this is a step that is going to be really difficult to qualify and validate thinking about commercialization. That is the reason that we are moving from this density gradient into the full empty separation by anion exchange chromatography. No? With this new next generation process, we are now being able to produce 500-liter batches with two different serotypes, AB8 and AB2. The AB8, as you can see, we have around 10 times increase in the number of viruses, and then with AB2, we have around 40 times increase. In both cases, we are getting around 1E14 VGs at the drug product, means after purification per liter of culture. That for an ABA is really impressive, but for an AB2 that everybody knows that is a very bad producer is just remarkable. No? So the conclusion that I want you to think about is that we think that our fast scalability, we have a fast scalability from research all the way up to the commercial with high yields. Our platform approach minimizes the time and the cost for process development and validation. And we really believe that the access to our CMC package and data management simplify and de-risk all the regulatory assessment. So thank you very much, and I will take any question. Thank you.